The final step is the collecting duct. The collecting duct is permeable to water, and it's also permeable to urea, and its function is regulated by hormones, and is especially the antidiuretic hormone, which is pituitary hormone. So, as usual, we have our epithelium with the apical membrane and the basolateral membrane. And the antidiuretic hormone, but also vasopressin, which are secreted, as I already mentioned, by the pituitary gland. Um, they are secreted as a consequence of the increase of osmolarity of the plasma. So IDH binds the vasopressin 2 receptor on the basolateral membrane of the collecting duct epithelium. This is a metabotropic receptor, which leads to the increased concentration of cyclic GMP. So it's a GS coupled receptor, which leads to the increase of concentration of synthesis of the second messenger cyclic AMP. And cyclic AMP drives the translocation of aquaporins from vesicles in the cytoplasm into the apical membrane. So the aquaporins are then inserted into the apical membrane, and what aquaporins are, as you can tell from the name, are pores that let water flow through them. Okay, so are fairly big holes in the plasma membrane, which allow the passage of urea and water. So ADH effectively stimulates the reabsorption of urea to cause the hypertonicity of the interstitial fluid. Okay, so the translocation of aquaporins on the apical membrane will allow the passage of urea from the lumen into the interstitial fluid. Leading the interstitial fluid to be hypertonic. And we stop here for now, because we will go into more detail on how the collecting duct and the ADH regulate the loss of water and of urea. For now, just remember that it is permeable to water and urea and is regulated by hormones such as ADH. I think you can see where I'm going with this, right? If ADH activates V2 receptors, which lead to increased CAMP, which leads to the translocation of aquaporins on the apical membrane, which are permeable to urea and H2O, leaving the lumen hypotonic and the interstitial fluid hypertonic because of the movement of urea, then water will follow. And if water will follow, it will be reabsorbed into the interstitial fluid and leave the urine more concentrated. But I don't want to spoil too much because it's a complex process, and I will go into more detail in a later stage. Okay, so to sum up, and I think it's very important that you have the general overview of the kidney function. It's complex, so you don't want to put too much into your brain at the same time. Okay, so make sure that you get it from now. Okay, just go through it in your head and make sure that you get what we've been talking about so far before we go into speaking about more detail about the regulation of potassium, sodium, and water. Okay, so to sum up, sodium is reabsorbed 99%, and 66% of this process occurs in the proximal tubule, 20% in the loop of Henle, and 12% in the distal collecting duct. Also, potassium is reabsorbed at 99%, 66% in the proximal tubule and 33% in the loop of Henle. Only 50% of urea is reabsorbed, but these can vary, and largely in the collecting duct. Water is 99% reabsorbed with an excretion rate between 0.4 and 2 milliliters per minute, depending on the ADH. In the proximal tubule, obligatory secretion and reabsorption occur of glucose, amino acids, sodium, and bicarbonate. In the descending loop of Henle, we have the concentration of urine, while in the ascending limb, we have the reabsorption of sodium and chloride with the concentration of urine. In the distal tubule, we do have the regulated absorption and secretion of various ions, including sodium and potassium, while in the collecting duct, we have the concentration or the dilution of urine depending on the secretion of ADH, which depends on the osmolarity of the plasma. Now we go into the fine regulation of sodium balance. Okay. First step, the renin-angiotensin-aldosterone system, which you probably have already heard of in the regulation of the blood pressure, because remember, everything is connected in the human body, is activated by low sodium concentrations in the distal tubule. The renin-angiotensin-aldosterone system is activated by low concentration of sodium in the distal tubule. Not only by that, it is also caused by a decrease in blood pressure, so by the collapsing of the arterial walls of the afferent arterioles. Okay. And the two things are absolutely connected, because low concentration of sodiums in the distal tubule and reduced blood pressure are two related phenomena, because higher concentrations of sodium in the blood result in higher volumes of water in the plasma and increased blood pressure. So reduced concentration of sodium and reduced arterial pressure are two absolutely related phenomena. Renin is produced by the macula densa cells, yeah, these cells here. Probably I should do it in yellow. Macula densa cells. Macula densa cells in the distal tubule. Okay. Renin is an enzyme and an hormone which is secreted into blood. How does this work? Renin, so well, we have low sodium in the distal tubule, okay, as we mentioned, or reduced pressure in the afferent arteriole. In any case, these events lead to the activation of the macula densa cells, which activate the juxtaglomerular cells, which release renin. Renin is a protease, and as a protease in the blood, converts the angiotensinogen, which is produced by the liver, into angiotensin 1. Angiotensin 1 reaches the lungs, in the lungs there is an enzyme called angiotensin converting enzyme, which converts angiotensin 1 into angiotensin 2. Angiotensin 2 in itself is a vasoconstrictor. So it promotes the contraction of muscular arteries. But other than being a vasoconstrictor, it's an hormone which acts on the cortical cells of the surrenal gland. On the cortical cells of the surrenal glands, it promotes the secretion of aldosterone. Aldosterone is a steroid hormone which acts on the distal tubule, promoting the reabsorption of sodium. How does this occur? Aldosterone functions by increasing the insertion of sodium channels onto the apical membrane. Now remember, the bilayer of phospholipid is absolutely impermeable to sodium. So you need to put channels in it. The higher will be the quantity of channels present in the plasma membrane permeable to a given ions, such as sodium, the higher will be the permeability of the plasma membrane to that ion. So aldosterone acts on the epithelial cells of the distal tubule and of the collective duct and promotes the insertion of sodium permeable channels on the apical membrane. But that in itself wouldn't be enough, because once I have the hole in the membrane that allows the passage of sodium, I need energy to move sodium from the lumen into the interstitial fluid. So I need a gradient, since sodium is both a chemical and an electrically charged species, an electrochemical gradient. Now, this electrochemical gradient, as we mentioned, is generated by the ATP sodium-potassium pump, which is always there in the basolateral membrane. But in presence of aldosterone, the amount of sodium-potassium ATPase pumps located in the basolateral membrane increases. So aldosterone has a double action on the epithelial cells of the distal tubule and of the collective duct. It increases the amount of passive channels for sodium on the apical membrane of these epithelial cells, while it increases the number, the density, and the total number of the sodium-potassium ATPase pump on the basolateral membrane, increasing the amount of sodium that is actively pumped from the cytoplasm of the epithelial cells into the interstitial fluid, hence generating the electrochemical gradient for sodium, which drives sodium to move from the lumen into the cytoplasm and from the cytoplasm into the interstitial fluid. Now, this will generate a transepithelial potential. This transepithelial potential can vary between -5 and -70 millivolts. And of course, the more sodium will move into the interstitial fluid, the more negative will be this potential. If we put our reference electrode, of course, into the lumen, okay? Because positive charges will leave, the lumen, okay? So here we're talking about transepithelial potential, okay? The potential difference that there is between the lumen and the interstitial fluid. Remember, not the plasma membrane potential. The transepithelial potential is a different thing. So because of the generation of this transepithelial potential, then we will have a driving force for potassium. Now, we do have leak passive channels for potassium, both on the apical membrane and on the basolateral membrane. And so depending on the direction, depending on the intensity of the transepithelial potential, potassium will move into the two directions. So in presence of aldosterone, which increases the reabsorption of sodium, as sodium moves from the lumen to the interstitial fluid, driven by the increased sodium-potassium ATPase pump on the basolateral membrane and increased passive sodium channels on the apical membrane, we will have the accumulation of positive charges in the interstitial fluid, pushing potassium to move back into the lumen. So aldosterone causes sodium reabsorption and potassium secretion. Aldosterone causes sodium reabsorption and potassium secretion in the distal tubule and the collecting duct. We can see it. We can see it in this graph. The amount of sodium excretion from the moment of the infusion of aldosterone in the blood of a person. So this sodium excretion, so the concentration of sodium in the urine in micromole per minute, you will see that from the moment of the infusion of aldosterone, so within just over an hour, the levels of sodium in the urine will start to decrease. And this is mirrored by a progressive increase in the excretion. So let's go through these again, because this is very important. Very, very important. It is complex, but it's also very rational. So in a way, easy to understand because it follows rational principles. Let's start from the secretion of renin. As we said, the secretion of renin is caused by a decrease in renal perfusion, which is, corresponding to a decrease in the arterial pressure in the afferent arteriole, or by a decrease of concentration of sodium in the collecting duct. These two events, which correspond to a decrease in renal perfusion, lead to the secretion of renin from the macula densa cells. Well, the glomerular cells stimulated by the macula densa cells. And renin converts angiotensinogen, which is a protein secreted into the blood by the liver, into angiotensin 1. Angiotensin 1 in the lungs is converted by the angiotensin converting enzyme into angiotensin 2. And angiotensin 2 does a lot of things. One of the two actions, well, two of the actions that we have already mentioned, is the vasoconstriction of muscular arteries and arterioles. So by, in itself, leading to a midterm, so to speak, increase in arterial pressure. So angiotensin is not as fast as an autonomic response to the change in pressure detected by the aortic arch or the carotid sinus. So it's not a short-term regulation of blood pressure, but it's definitely mid-term, okay? Because it acts as a vasoconstrictor on the arterioles as a consequence of this downstream cascade of activation from renin to the generation of angiotensin. But it also acts on the cortical cells of the surrenal gland leading to the secretion of aldosterone, which leads to the reabsorption of sodium and water, and yes, to the tubular reabsorption of sodium chloride and, of course, water retention and potassium excretion. It also works on the sympathetic system, and it increases the sympathetic system. And last but not least, it also promotes the secretion of antidiuretic hormone from the pituitary gland. And as we will see, the antidiuretic hormone, well, we have already mentioned it, but we'll see it in more detail. The antidiuretic hormone leads to the concentration of urine and reabsorption of water into the collecting duct. So by reabsorbing water, both via the aldosterone system and the ADH secretion, we are increasing the volume of plasma and of the interstitial fluid as a whole, and so we're increasing blood pressure. But this is a long-term regulation of blood pressure. So you see how beautifully regulated systems are. I am really fascinated by the ability of the body to, yes, respond very quickly to a decrease in blood pressure via, the aortic arch and the carotid sinus. I mean, I signpost you to the lectures on the regulation of cardiovascular system and of the blood pressure. So as the short term is the sympathetic regulation of blood pressure, and it's almost immediate. To the mid-term regulation, because a decrease in blood pressure results in a decrease in renal perfusion, which leads to the renin-angiotensin system activation. And then angiotensin itself is a vasoconstrictor, so it's slower, of course, than an autonomic response, but it's not as slow as aldosterone and ADH. It's a mid-term regulation of blood pressure by increasing it, and then we have this long wave of regulation via pituitary gland and aldosterone. Now, these systems are not on and off, so to speak. These systems are always active. They're more active or less active. Okay, so it's a continuum, it's a spectrum of activation. You never have zero renin or yes renin. Okay, you have variable levels of renin in the blood depending on how the overall average blood pressure changes in time. So it is this very refined, very sophisticated feedback loop that keeps the blood pressure absolutely spot on its homeostatic levels, of course, in a healthy organism. This is how beautiful our organism is, how sophisticated it is. I don't think we have machines sophisticated enough to do something like this. It's truly a marvel. So in terms of what stimulates the renin-angiotensin-aldosterone system, I've already mentioned it, but I will mention it again. It will be a decrease in the sodium concentration in the distal tubule, a decrease in the blood pressure, and a decrease in the blood volume. Of course, these three events are highly causally related with each other. Okay, they're not independent. Okay, a decrease in sodium ion concentration and a decrease in blood pressure and a decrease in blood volume are highly related events. While the renin-angiotensin system is inhibited, so it receives a negative feedback by, on the other hand, increased blood pressure, increased blood volume, and constriction of arterioles, for example, due to the action of the sympathetic nervous system. So to sum up the regulation of sodium, sodium is obligatorily reabsorbed at 66% in the proximal convoluted tubule, then 25% in the thick ascending limb, and then regulated reabsorption occurs in the distal convoluted tubule, and again regulated reabsorption occurs in the collecting duct. the excretion of sodium is always less than 1% because we dearly need sodium to survive. Now we go into potassium. This lecture is a little bit longer than 20 minutes. It's actually much longer than 20 minutes, but you can press pause if you are tired. So if you're tired now, press pause and go have a cup of tea and continue. I decided to keep it together because the regulation of sodium and potassium balance fit well together. So if you're tired, just press pause and go for a cup of tea, and we will start after that. Actually, here I am back after a cup of tea. You would have never thought if you continued to watch the video, you know. But yeah, I could also press pause for recording this one. Okay, so as you remembered, probably because we just mentioned it, aldosterone has a double action, right? It promotes the reabsorption of sodium and it promotes the excretion of potassium. So let's look at that in more detail. So first of all, some terminology. The excess of potassium is called hyperkalemia. Okay, so hyper, above, kali, kalia, I don't remember, ancient Greek for potassium, and emos means blood, okay? So too much potassium in blood, okay? Hypokalemia is the deficiency of potassium, the reduced concentration of potassium in the blood, okay? Hypo means under, kalia, and I need to check this etymology, means potassium, and emos means blood, okay? Hyperkalemia, hypokalemia, you got it. So aldosterone promotes the excretion of potassium by, guess what, increasing the number of potassium channels in the luminal apical membrane to increase the potassium secretion, yeah, by the principal cells. It also increases sodium reabsorption and the sodium-potassium ATPase activity and expression on the basolateral membrane, as we said. And this leads to an electrochemical gradient across the luminal membrane, which we call the transluminal potential. So those factors that increase the gradient will increase the secretion of potassium, okay? While the factors that decrease the gradient will decrease the potassium secretion. And also, last but not least, the dietary potassium. If you eat more potassium, you will have hyperkalemia. If you don't eat enough potassium, you will have hypokalemia, okay? So we've listed three main factors which allow to regulate potassium, okay? One is aldosterone, which, as we said, promotes the excretion of potassium by increasing the number of potassium channels on the apical membrane of the epithelial cells, of the principal cells in the distal convoluted tubule. The electrochemical gradient across the luminal membrane in the distal convoluted tubule, and the dietary potassium. A fourth factor are the disturbances in acid and base balance in the blood. Why? Because in the kidney epithelia we have sodium and potassium exchange. So an increase in the pH in the blood causes hypokalemia, okay? Causes the loss of potassium out of blood into cells. While the acidosis of blood, so a decrease in the pH of the blood, causes hyperkalemia. So an increase in the concentration of potassium in the blood, so potassium moves into blood out of cells. fifth factor which regulates potassium balance, and you need to know these all. I'm not joking. These are very important factors. Diuretics, drugs which promotes the loss of urine. Yeah. So diuretics often work by inhibiting sodium-potassium chloride co-transport, and that will result in the increased urine production. So the co-transport of sodium-potassium chloride to produce profound kaliuresis and blood hypokalemia. Okay. Also negative charges in the lumen, so the presence of large anions, so for example sulfate, I don't know, the ingestion of weird food. The presence of large anions in the lumen of the tubular system in the nephron, especially in the distal convoluted tubule and collecting duct, leads to the increase of potassium secretion. So let's look at it in even more detail, okay? So in the distal convoluted tubule, we have alpha intercalated cells, just epithelial cells. And the potential of the lumen is minus 50 millivolts, and the potential of the blood is 0 millivolts, okay? So the sodium ATPase potassium pump on the basolateral membrane on the right-hand side leads to the movement of potassium from the interstitial fluid, ergo the blood, into the alpha intercalated cell. But then we do have leak potassium channels selectively on the basolateral membrane. So if there is excess of potassium intracellularly, that will move outside of the cell. On the apical membrane, we have ATPase proton pumps, which move protons from the lumen of the cell into, from the cytoplasm of the cell into the lumen of the distal tubule. And also we have ATPase hydrogen potassium pumps, which pump one proton in the lumen of the distal tubule and one ion of potassium in the intercalated cell. So in these conditions, potassium is reabsorbed into blood. Why? Because the ATPase pump on the apical membrane promotes the reabsorption of potassium from the lumen into the inside of the cell. The ATPase sodium-potassium pump on the basolateral membrane will promote the movement of potassium from the blood or the interstitial fluid into the cytoplasm of the intercalated cell. But then the only leak channels that allow the passive movement of potassium from the cytoplasm of the cell into the extracellular fluid is on the basolateral membrane. So, this excess of potassium inside of the cell can only move out from the basolateral side, which means on the blood interstitial side on the right. So this is particularly true when we have low potassium in the diet. So something similar occurs in the late distal tubule / collecting duct. Really hard to, draw a border between the convoluting tubule, the distal convoluted tubule and collecting duct. So let's call it late distal tubule collecting duct. As usual, we have an electrical potential in the lumen of minus 50 millivolts and a potential in the blood of 0. But this point in the late distal tubule and collecting duct, we don't have the alpha cells we were talking about beforehand. We have other kind of epithelial cells. We've got principal cells. And the principal cells are characterized by passive channels for sodium and potassium on the apical membrane, passive channels for sodium and potassium on the apical membrane, and ATPase sodium potassium pump on the basal membrane, basolateral membrane. So we already know what that means for sodium. Okay, we already know that for sodium, this will generate a gradient which promotes the reabsorption of sodium from the lumen into the blood. But for potassium, this means an increased secretion slash excretion, right? Because the sodium ATPase pump brings three ions of sodium from the cytoplasm of the cell into the interstitial fluid, but 2 ions of potassium from the interstitial fluid to the inside of the cell. And because of the selective positioning of the leak channels of potassium on the apical membrane, then potassium will move from the blood into the principal cell, into the lumen. Now, aldosterone increases this process. We've seen how aldosterone increases the amounts of sodium channels on the apical membrane and of the ATPase sodium potassium pump on the basolateral membrane. And that corresponds to an increased reabsorption of sodium. But it also increases the amount of potassium which is transported from the blood into the principal cell, and because of the passive leak conductances to potassium onto the apical membrane, the amount of potassium that is secreted from the blood into the lumen and then finally excreted. That is how aldosterone promotes the excretion of potassium. So, to sum up, most of potassium is obligatorily reabsorbed in the proximal convoluted tubule, 67% of it. Then 20% of it is reabsorbed into the ascending limb of the loop of Henle. In the proximal convoluted tubule, low potassium diet leads to the reabsorption of potassium, while in the late distal convoluted tubule and collecting duct in the principal cells, both high dietary potassium, aldosterone, acid base, flow rate, and luminal anions will lead to variable secretion of potassium via the modulation of the sodium potassium pump on the basolateral membrane and the selective presence of leak channels for potassium on the apical membrane. The excretion of potassium is extremely variable, and it goes from 1% to 110%.